Hello. Morning. We'll wait for a few more people here. There we go. Welcome, Trevor.
Thank you. Good morning.
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 All right. So yeah, we welcome to meeting. We didn't have a open source meeting yesterday, but we did have a DivaWorm meeting last week. So DivaWorm, we covered a number of topics, kind of in C. elegans molecular biology, talking about CompuCell 3D. One of the people who attend the meeting regularly went to the CompuCell 3D workshop, and then we talked a little bit more about modeling. And so it was a really nice set of discussions. So we didn't have our cognition futures nor our cybernetics meeting. We'll have to pick those up in a few weeks. So we'll see how that goes. Okay, any updates?
I'm not quite ready, but I've got some updates.
Okay, yeah. Yep. We'll come back to that later. Hussein or Trevor?
I sent you that message. The idea I was thinking of, unfortunately, I started delving into literature. I actually spent a few days and it turned out to a dead end. I've got another idea that I'm kind of playing around with. It's still gestating, so I'm not quite sure if it's ready for public dissemination, but that's me. Yeah.
Well, yeah, that sounds, it's sort of that process of identifying a problem and seeing if it's viable. And you don't want to spend time on it when it's not viable. So, that's something that we can talk about. We don't have to do it within the next week, So,
for sure.
I'm
just going to ask out of curiosity, do you have many like experimentalist collaborators that I was just curious.
Yeah. We have in the past. We don't. It's kind of hard to get a consistent collaboration there, but that's something we could set up if that's. Certainly we'd have to figure out how to do it. So.
Yeah, I was just curious. There's I kind of have an idea in the realm of like sonogenetics. I'm not sure if you're familiar. It's just using ultrasound to like trigger neuromodulation, and I think there are some molecular opportunities for maybe making better tools. And so, but anyways, yeah, we can talk later this week. I don't know if that if something like that would be of any interest to you.
Yeah, that would be interesting. Definitely.
Yeah, definitely. That sounds interesting.
Okay, cool. All right. Well, that's positive. I'm glad to hear that. Yeah.
Yeah, please, make sure that we. I don't want to force you to design a formal set of experiments before we talk about it. So, if you're at a point where you think that it's worth kind of writing up a 1-page or 2-page description, then that would be great to look over and talk about.
Awesome. Okay. It's a tricky thing because of course, the most tantalizing things require experiments. So it's like the most that I could do without experiments is just like a lit review, but perhaps there's still value in something like that.
Yeah. Yeah. Well, you have to just decide what you want to do and what the questions are before you can get to how to implement it. That's usually the case. So, there's some experiment we can design, but we have to know kind of what we're looking at and what kind of, what we want to employ to get the data that we want. So.
If I may ask, I know I've heard like this talk of organoids going around. What's all that about? Does someone work with organoids in here?
Well, Morgan is attempting to set up an organoid collaboration. So I've been interested in organoids for quite a while, and Morgan is actually working on doing some wet lab collaboration. So Morgan, did you want to say more?
Yeah, that's one of the things that we're setting up here in San Francisco. But we're still in the process of getting our wet lab space up and running. So hopefully by next month that will be the case. Yeah, we're supposed to have our open house. I think the 20. Oh, I don't have a calendar in front of me, but the 22nd, coming up.
Yeah.
But it'll still be some time from that, right? So organoid culture, when you look at like the kind of cerebral organoids culture that we're hoping to do, still has some infrastructure requirements that we're not quite at yet. But I should be visiting UCSC pretty soon. And there's a Braingineers, so braingineers.ucsc.edu. You can find the work of it's like I don't know how to describe it, a subgroup in the Institute of Genomics. A special interest group. I'm not sure formally what the Braingineers are, but definitely like a bio-E component too, as well as it's like a bio-E group together with some more developmental and genomic groups. And the nice thing about the Braingineers versus say some of the other like UC San Francisco or Stanford groups is that they make all their lab automation hardware. They're publishing that plans and designs for all of that hardware as well in an open source fashion. So that gives us, with an eye to, as you see with lab automation, with an eye to scalability, right? And with an additional eye to making cloud-enabled research, because which is really separate from the lab automation. But one of the PIs, whose name I forget right now, has a lot of M's, is from Bolivia. And so one of the things that they've done is make all this hardware very, very automated, but then they've also made it cloud accessible, so that students in Bolivia, where they don't have access to this kind of wet lab infrastructure, are able to do their own experiments. So Bradley and I have had at least one Jitsi call with Katerina Wojtek, who's the first author. I mean, she's finishing up her grad school PhD. She's the first author on this kind of open source hardware infrastructure for organoids. Really interesting group, and at the last Celsius open house, I got to meet someone from the UCSC's innovation group. So I think in general that's the group that's kind of marketing the university's IP. In this case, it's all open source or publicly available stuff, but still, they're very keen to help raise profile of any of the university's work, and so we're definitely getting some. Elliot Roth is another principal at Celsius, and so we just got a $5,000 grant for some of the infrastructure costs too. In addition, a lot of it's 3D printable, and so I'll drop a link in the chat for you today, Trevor.
Thanks. No, I appreciate it. It sounds very impressive. Is there a particular focus? Sorry, I know this is probably premature.
Oh, so what are we? Yeah. So when I think about what we're like specifically targeting in terms of the science at this point, brain engineers have covered a lot of what we do want in terms of we want a lot more automation and a lot more scalability in terms of culture. But the real focus is on electrophysiology of the organoids. And so in terms of a protocol, so one, that is a protocol, or at least they've done this for some rat cells at UCSC, but the protocol that we would hopefully be able to reach is from San Diego's Allison Moutry, and peripherally, I mean, in terms of the electrophysiology analysis, Bradley Wojtek down in San Diego. But just this spring, Allison, or Moutry Lab published, I forget what did they call it, a semi. Blanking on the exact title, but it's something funny, like semi-automated protocol, or I forget. Yeah, it's something like semi-guided. Like I've never heard semi attached to this particular word, but there's they have put together a kind of published a protocol in terms of electrophysiology, and one of the kind of not, I don't want to say controversial, one of the surprising findings of the early Moutry Wojtek work had to do with the power spectrum of the electrophysiology, the organoid. After like 80 days, so close to 3 months, and perhaps even longer. But what Madeline Lancaster has pointed out, one of the ways in which you can really one of the ways that you can really mark a change in the organoids is by its network dynamics, and as she points out, it's when the organoid is starting to behave, or when the multicellular dynamics are starting to behave in real network ways, is to her is the sign of what what you're really trying to achieve, right? So there's just a number of questions about changes in kind of information theoretic measures, network complexity, etc., that we can track with good automated electrophysiological recordings, and that's like our very modest goals at this point. It just to some degree just replicating that work, and then the work of some other groups that are related to that. So then there's like a 20. The first paper was like 2018 paper with Motri Wojtek, but then there was like a 2020 paper that came out of Santa Barbara, and that had Shrikatan Nagaraj, who's the MEG director at UCSF, in terms of the electrophysiological analysis. Anyway, just broad strokes on that.
If I may ask, when you say electrophysiological analysis, are you talking like patch clamp, or are you talking like calcium imaging?
No, I mean that would be like, so we are now, or I should say, like this recent discussion I had with a group in Bristol, but this is MEAs, you know. So just electrode arrays that and so one of the interesting, or one of the questions we have in terms of being able to do these kinds of long-term recordings is you know, do you grow on MEAs? Are you able to manipulate organoids to move them around in some sort of you know, good mechanical fashion? And yeah, a number of things about doing those recordings that again are brought up in Katerina's paper. That this is her biorXiv paper. So this is the yeah, currently unpublished, but on the archive. And they're using it's the one device that is still, you know, commercial. So it's Maxwell Biosystems, I think, MEA.
Yeah. Thank you for describing that. That sounds interesting.
Yeah, and they've got some preliminary data, or you know, they've got some example recordings. So again, like her, you know, it's been updated since then, but it's like first hit archive like March 17th. But anyway, I'll drop some links in just a minute.
Yeah, yeah. Thank you for that update, Morgan. Organoids. We have a brain organoids channel too in the Slack, so it's brain-organoids, and we've put a lot of things in there, a lot of things to read over, and so yeah, that's like, you know, we're really kind of trying to figure out how to do this and how to use this to answer some questions. In the meetings, we've, you know, tried to look at different papers, protocols, but also, you know, we looked at the one where they were playing video games with a organoid. I can't remember what they called that.
Well, that was Dishbrain.
Dishbrain, right? So yeah,
that was 2D culture.
Yeah, 2D culture. So yeah, there are a lot of things that one could do, but it's just seeing what, you know, what's out there and trying to find things that we can do, given our resources, and yeah. Okay, so looks like Hussein was here. I don't know if he left, if he is going to come back, but hello, Hussein. Hello, Sarah and Jesse. So yeah, I don't know. Oh, hi. I don't know if you want to give an update, Sarah. You ready?
Yeah, sure. Yeah, so basically, like, I'll just share my screen. So, yeah, like, I just thought today I'll kind of like, I've been working on a few small things, and I've submitted like 2 pull requests as well. So, like, this was the first pull request, and this is kind of the graph that I have so far, like 2 metrics that are being graphed for the project. And so you can go through this pull request, anybody who's interested, and then another pull request that I just submitted regarding the issue creator agent. So I just thought I'll give a few like kind of demos on what I'm like working with and how it kind of looks. So the first thing that like I was doing last week as well, and then I completed this week, was the issue creator agent in the open source sustainability project. So basically, like even last time, I think Bradley had this thought like, basically, I'm just trying to use an LLM to create more issues so that the more issues there are, the longer, the more time steps we have in our simulation and the longer our simulation can run. But then one thought was that what if it just kind of creates some garbage? And another thing that I had nagging was what if it kind of just duplicates issues that I already have? Because both of those would be bad. Generating garbage would be bad because you couldn't put any good code to that, and that would lead in like bad overall metrics. And if it basically if it does something that existing issues already have, that would be bad because it would kind of conflict with like the code would be there already, and then autocode rover would not know what to do, so it would not be able to generate proper responses. So both of those things would be bad, which is why I kind of made this issue creator agent in the way that it generates everything systematically, like step by step. So instead of just telling it to generate a whole issue, the first thing I do is parse the existing issues, like whatever existing issues I have, I take them. So for example, like I had run this command with this command line argument of 4. So I already had 3 issues because I already have 3 issues and I want to run the simulation with 4 issues. The issue creator agent tries to create the one remaining issue. So the first thing it does for that is it checks all the existing issues and it parses like the basic title and description that these issues have. Like it takes the first few sentences of each of these files and it stores that here. And what this does is these existing issue titles are given to the first LLM call, which is just to generate the issue title. So it is just told that these are the existing issues and you have to create something that does not conflict with these. And it's also given the existing code from the Python file. So based on that, it comes up with an issue title. So this is the issue title that it came up with, which was to implement the modulus operator functionality in the calculator class. Once this issue title is done, that is given to the next, the title is given to the next LLM call and it is told to create a description for this. So it makes this basic description. Then that title and description are given to another LLM call that generates some basic example code. Then that is given to another LLM call that checks like basic, that says like what other resources did it check. And that is given to another LLM call that generates some system info for where this issue was checked, etc. And then all of this is combined and given to an LLM call that creates the final issue. So up till here, like, and that everything is combined in like this way, I can like be sure that it's not generating gibberish because it was given prompts for each and everything. And I can also be sure that it's not going to conflict with the existing issues. The human in the loop can still be added, but like it is not a necessity for now. Like it's generating some pretty good responses for now. And then this is like just the rest of the simulation without ACR invoked. So it's just like without the actual code solving happening, a basic this. There was also this code quality thing that I added, where it basically like rates the kind of gives a review to what the code pull request was, and based on that it parses a code quality. And yeah, there was another demo that I had going on. This is what happens, like right now I ran this with issues 4, and I already had 4 issues generated. So this basically just ran on its own. It didn't generate any new issue. It started solving these issues, did the LLM's bidding part, and started the autocode rover in the background, which is actually solving the code. So this is everything from autocode rover, and like this is still working. So this I wanted to like I have another one with this completed, but the graph of it got lost. So basically, like even while this is generating, the graph keeps on building. Like once the first time step is done, the metrics for the first time step will show up on this graph. So that is what I wanted to show, and like how it's like all coming together now. And then another thing that I'm working on right now is the personalization part. Basically, like for each agent, whether it's a contributor agent or a maintainer agent, giving it like some basic storyline. So currently I'm working on currently I've done where I've created a system message for that agent. Basically, the system message gives it like the system message is what is given before any LLM call is made to this particular LLM. So this is, and it is like appended to every LLM call. So basically, the system message is going to help it remember like what its experience is, why it is like why this agent is there in this particular simulation, and hopefully like I'll see better results in the metrics and even in the bidding part, especially because of this personalization once it's completed and integrated. So like now I'm just going to focus on trying to get this personalization and some other stuff done, and then focus on a bit of the documentation. Like I have everything documented till now in the terms of like readmes and logs. But I'll try to like get everything accumulated at one place now, and all the pull request links and everything like that.
All right, that's great. Thank you for the thank you for the update. It was great. Yeah, I wanted to see where you were. I think I mentioned that as we come up to the end of GSOC, which is August 26th, that's the deadline. We need to kind of find a stopping place. So if we can find a stopping place where everything is, we can wrap things up and get something approximating a submission to Google, then that's what we want to do. So, you may have things you want to do, sort of, there are a lot of things you want to do to your project, right? I would find a stopping point where I kind of stop what I'm doing, make sure it's working, and then make sure that the documentation is sound, and then make your submission from that. And then later on we can work on more details. So they just want you to find like a stopping point for the project. I think I like a lot of the stuff you're doing. I think you're making some progress on kind of you have the framework, and then you're kind of finding ways to make sure that you can verify what's going on. So that's good, because of course if you just generate large language model text, it might just be like, you might get duplicates, you might get garbage, etc. So having that sort of, those checks that can be implemented is good. And, we don't have to we don't have to make like this huge elaborate solution. It's just something that will kind of give us a way to do that. And we can maybe, next year or after the end of the project, we can come up with better methods for it. So I wouldn't worry about that too much, just to have something that's working.
Right, definitely. So yeah, in the next week I'll try to kind of do, like as you said, get to a stopping point and just integrate everything, and then after that like I'll just stop actually and just focus on the documentation, because that's definitely there where like I always feel like there's something I can add. So yeah, for now I'll just get to like a good enough point and stop it.
Yeah, it's good.
Temporarily till we can just like finish all the documentation and get it all done and then come back.
Yeah. And you're welcome to continue working on the project if you want to after the end of GSOC.
Yes, definitely.
Yeah. This looks pretty
Definitely.
good.
There's like a lot of things that I had that I kind of wanted to add, there was a Sareji thing that I'd explored right in the beginning. It's kind of like now there's not enough time and not enough scope to integrate that for the GSOC period, but that's definitely something I want to try to integrate even later, and even like other ways that LLMs could be used. Like I remember you mentioning Bratenberg vehicles like a very long time ago. So I've not been able to look at that while I was working on this. But definitely later, once the GSOC period is over, I would like to work on it.
Yeah, that'd be good. And of course, what you can do for those things is, kind of sketch out what the issues will be for that. So, you don't need to, that doesn't need to be your first priority, but to just make notes about what the issues should be. And then, we'll populate the project board that we have been using for the open source meetings. We can put those on there. And, then you can either address them later or someone else can address them, just so that we know what the open issues are, where we might want to improve
Right,
it.
definitely. Yeah.
All right. That's very good.
Yeah, I'll do that as well.
Okay. All right. Thank you, Sarah.
Thank you.
So we're coming along on GSOC. We're kind of coming to the end of that at the end of the month. So I actually, for DivaWorm, my 2 students there, I've gotten an extension for a couple more weeks. So they're working on things as well. Their projects are a little not as far along as Sarah, so I'm giving them an extension. So, but that's the same process. When you get to the end of your project, you want to prioritize the things that are doable, maybe want to have, maybe are kind of reaches, and you do the ones that are the easiest. You finish that up, and then you make issues for the things that you would like to have, or that are reach issues, and those are typically things that you can do later, someone else can do later, as long as those things are codified in the project space. So we'll be coming to, this will be fun. I think they're all very good GSOC projects, and I can't wait to see the sort of submissions. And I think we've had a lot of nice demos this summer of different types of things that people have been doing.
Hey, can you hear me all right?
Yeah.
I'm good. It's been a good week, mostly busy times. I'm curious as to what else we wanted to talk about today, so that might affect some things that I do or don't talk about. But for just very general updates, just a lot of things for JoPro have been coming along. I'm just doing major, I don't know, just call it an infrastructure update. And like a byproduct of that is sort of the website. And I mentioned in this chat here, I said to Morgan, like, if you want to talk about like getting, I don't know if there is anything written up like for what, when we say brain organoids, we don't like, what does it mean? Just a Slack channel. But like, if we want to, I don't want to say formalize, it might be a scary word at this point, but basically make like a project description about what is trying to be done and where it's at. Like, that's something that I'm interested in helping out with. I mean, I've been in the discussions for a while anyway, but more like I'm doing a whole lot of that recently and trying to kind of make things a little more tenable to both, it's this fine line between like administrative work and functional project management and general clarity of like what is happening. So that's the last 2 weeks, maybe even 3 weeks, I've really been focusing on that stuff. And this week has been exciting because I'm really starting to reach out again to many, I guess, I don't know, potential former and potential partnership collaborators. We've done some things with ULB in the past, and I'm kind of following up there again. I have some plans to go to UC San Diego, where I'm currently a student, but I'm basically remote. But in September, there's a few events I intend to be going to there, and I don't think I'm going to get any like UCSD intern students this fall, but I think in the future and ahead, that is more of an option. There's a lot of things happening in the Boston area, so what will bear fruit and what will the fruit look like? We don't really know yet, but things are happening, and it's cool. That's the shortest version of my updates. There's for NICWIC, which is the New York Celebration of Women in Computing event. The update on that is we're pretty close to selecting a venue. It's kind of between a few of the bigger, like not New York City, but a few of the upstate cities we're looking at. And I'm excited because I think this is the year that we have enough support. As we all may know, if you're one person trying to make a change at an institution for how things are working, it's tough. But if you get an ally or two who are interested and able to make that change or invest in doing it, that's a big ball game changer. So I think this year will be really good for like building a little more community around NICWIC, and then also specifically there's some mentoring projects that I think fit well with what we've done in the lab, what we're trying to do, and what I'm trying to do with JoPro. So that's exciting. We had some meetings with the mental health working group today, yesterday, and also today, and some other like just JoPro admin stuff. We're fortunate to have a kind of like a formal, fancy, not formal, an informal but fancy in-person meeting here in Boston, and it went well. So that's the short version of it. There's also one other thing I'm really interested in trying to do. Some other like opportunities, I guess, or things to keep in mind are for people who want to be for those interested in NeuroMatch, which is sort of the, I guess, former conference series and current NeuroMatch Academy Summer School. This is the first time they've done certain courses. They have a new Neuro AI course that just wrapped up. I was a professional development mentor. I think Bradley was more of a full mentor for the projects. Yeah. But has ended. But there's this thing called Impact Scholars, which is sort of a continuation. It's the thing that everybody always wanted, which was, oh, I love my project, love NeuroMatch, but can we do a longer, more drawn out thing than just 2 or 3 weeks crammed? And that is Impact Scholars, from my understanding. So there's if you were a student, you kind of get the option to continue your project through, I think, March of next year. But there's also a big call for mentors again, because mentors will meet, I think, somewhat regularly. It's sort of like part-time job though, so it's a commitment. But they need mentors to help facilitate the projects and things like that grow. So definitely look into that if you're interested. I'm not sure I'm really qualified. I am for like everything that's not technical. Because I don't think I would be a good it's so funny because when I was a project the professional development mentor one of my mentees was like this sort of tenure track assistant professor, and I was like, well, I'm not quite there yet, honestly, in my computational neuroscience capabilities. But like in terms of other things and navigating spaces and just project development, and that I feel like, okay, I could do that. But so it's sort of like, it's a little bit unclear. I might ask them a bit more, like, are you basically looking specifically for like the mentors for the projects to continue on and get paid for it? Or is it like a little bit more inclusive of the professional development lens? I don't know. But either way, NeuroMatch like is something to check out. And for those who don't know anything about them, I'll just say they're very progressive in terms of how they're trying to make education accessible. And they're just a fun, innovative space to see what's going on. And they do some cool stuff. So check that out. Similarly, my last sort of general update is there's always, kind of events and things popping up. But one of them I'm particularly interested in is sort of a workshop. I forget who hosts it, so this might not be the most useful. But there's an upcoming potential workshop for, I think they're calling it something like, it's not focused research organization, but it's something along the lines of a research organization that's in between academia and industry and isn't necessarily like somewhere in between those conventional scopes. And I'm hoping to attend that. I don't know if I'll get accepted to it, but I think it would help. Because, if you don't know by now, now you know, but like we're in like this sort of interesting para-alt academic space. And there's a lot of things in that space, but sometimes having, the structure about what those are, how do you do what part or what development of the research path, or like pushing an idea or theory, or the outreach of an idea or theory, whatever, like what do you do with that? So I'm been looking a bit into these I don't know, things like that, and there happens to be a workshop around such that's happening. Both one's in Boston, one's in San Francisco, one's going to be virtual, and I can't remember the name at this time. I think it was related to speculative technologies, but I don't know if it's them directly. But suffice to say, there's some interesting stuff happening in that space, which fits well with at least some of what JoPro is trying to do or facilitate. JoPro not only as a sort of mentoring branch, but also like the sort of project coordination, incubation, and development aspect. So, and our unique, let's say, take, aka, what can you do without a lot of funding currently that's still useful? So yeah, that's a very broad swath of things, but I'll leave it at that for now.
All right, that's great. Yeah, there are a lot of things there. Looks like you're investigating some of these spaces, seeing how we can implement, different mentorship programs and different, ways to sort of kickstart projects. So that's great.
Sort of, if I could add to that, something that Jesse has at least expressed some interest in too, that it would be awesome to get his help with, which is this upcoming NSF RFP on informal STEM learning.
Right.
And it seems very, very, very well aligned with both, Neurotech X and Celsius, which are the groups that I kind of originally was with in terms of considering that, but is actually super well aligned with orthogonal, right? and I would still, not to push it, but it'd be great to potentially, formalize orthogonal so that it could be partner on something like that, because I just think, this is kind of a unique opportunity in terms of kind of making a case for informal STEM learning.
Yeah.
And the kind of the institutions that support it.
Yeah.
And one of the things about this particular grant is that they encourage groups to do submissions with multiple organizations. So like if we can find overlap between lots of groups, like they were particularly encouraging of that kind of overlap, that kind of coordination in this particular proposal. Just to mention that too.
Yeah. Well, that's great. Yeah, and maybe we'll look into that more in the fall as well.
Sure.
So yeah.
Sure.
I think it was last week. Bradley and I met a little bit to talk about future plans and stuff like that. But I don't know if I don't have anything for you to say yet about either the model building stuff or the project management course. But I'm curious about those. I'm happy to do what I can with any of those things. I think the project management one is in spring, but those are in my head right now, so I just wanted to mention them.
Yeah. Well, I mean, I'm still moving forward with them. I'm just trying to, I'll probably work on it the next few weeks. So, yeah. And, I need to get materials together, probably kind of come together as I'm doing it. So, but yeah, I'm going to get things together in the next few weeks, at least to start off with, and then we'll go through. It'll be more the model building will be more interactive than the project management. But what I mean by interactive is just kind of having it's going to kind of be self-guided, I guess. But, okay. So yeah, I don't know, Jesse, I don't know if you had more to say or something. I'm going to do a few things here, and then we'll maybe come back to whatever you wanted to do.
Yeah, not really. If something comes up, I'll let you know, but you're in the clear.
Okay. This is something that Morgan attended, I guess. This is Cognitive Computational Neuroscience. This was in, I guess, is this in Boston or?
It was in Boston. Unfortunately, I did not make it.
Oh.
Yeah, so I got sucked into some work here, but all the videos, they made all the keynotes, all the talks available online, which is awesome.
Oh yeah, this looks great. So yeah, sorry you couldn't make it. Yeah, I know it's kind of hard, but if you have all the talks, then it's really interesting stuff here. So. Comparing artificial and biological networks, reconciling the dichotomy between Sherringtonian and Hopfeldian views on neurocomputations, varieties of human-like AI. We had a couple of keynotes with fireside chats. They had some contributed talks. So it looks interesting. What makes representations useful? So a lot of the things we've been talking about in this group, and we haven't attended this, but interesting stuff. Did you want to make any comments about it, Morgan? Any other comments?
So yeah, it's wonderful keynotes, and the what did they call them? General adversarial collaborations.
Right. Yeah.
is their kind of famous thing. So I haven't. So far, I just caught the Russ keynote, and just haven't dived in. The Neuro Academy was also overlapped with this.
Okay.
So that's the 2-week before NeuroMatch, there was NeuroAcademy.
Okay.
So that is also all online. That's from the UW Science Institute. So it's just a total, too much content this past week.
Yeah.
Together with something came up in terms of neurotech opportunity that I'm just trying to make something of for Neurotech X while it would be beneficial. So yeah. I got distracted and didn't make it to the East Coast. So the other thing that's going on right now is the Fedora flock to Fedora. So
okay, yeah.
Unfortunately, I did not meet with the NeuroFedora folks, which is too bad because they're distributed all over the world.
Right. Yeah.
So anyway, but sorry, almost home. We'll be able to sit down.
Okay.
Talk more appropriately.
All right. Yeah. Thanks for the update. This is something that I think came up in the Slack. I'm not really sure what channel. This is from Science Advances. And this is space as a latent sequence, a theory of the hippocampus. So, we have the hippocampus is well known for kind of, the different types of representations that exist in the hippocampus. People have probed the hippocampus in terms of memory, in terms of spatial cognition. You have different types of amnesia that have been studied in the hippocampus and the medial temporal lobe more generally. You have all these different types of place cells and grid cells and things that represent space, spatial landmarks and things like that. And so this is an interesting theoretical article that sort of proposes that spatial relationships are a latent sequence. And so what does that mean? We talked about, latent spaces in machine learning, and we know that that's where you store all of the possible outcomes of some data set. So you just sample the latent sequence and you see if you can find the correct representation. So this is Dalip George is on this paper. He's a very famous person in machine learning. Some other people on it as well. The abstract reads, fascinating phenomena such as landmark vector cells and splitter cells are frequently discovered in the hippocampus. So these are different types of cells that represent different aspects of space. So you can have landmark cells, landmark vector cells, these splitter cells that I talk about in the paper. And so, basically they proposed a bunch of different cell types that represent different things in this process of spatial cognition. Without a unifying principle, each experiment seemingly uncovers new anomalies or coding types. So, in case you haven't kind of figured this out, people have proposed a lot of different types of representations or cell types that contribute to these representations, but there isn't really a unified theory of this sort of thing. So place cells were first proposed in 1978, and there's been a Nobel Prize awarded in the last 15 years on grid cells and on some other types of aspects of spatial cognition and representation in spatial cognition. So this is people are very intrigued by this whole area. And so this is giving us a theory to guide us through this space. Here we provide a unifying principle that the mental representation of space, which is we have these representations within our brains that allow us to represent things in the world and in our brains and then operate on them. So we have this mental representation that they argue that it's an emergent property of latent higher order sequence learning. So sequence learning, of course, we know from language. It's very important in language. It's very important in procedural learning because you're learning sequences, you're learning procedures. And in this case, they're talking about this being important for spatial cognition because you go from landmark to landmark, or you go across the space, and you don't just, navigate the whole thing at once. You kind of navigate from point to point, where you kind of have to orient yourself and then navigate different, according to different orientations or, frames of reference. So it becomes a sequence learning problem. And of course, we have episodic memory that has been well characterized in the hippocampus, but we also have the sequence learning that this is something that they're proposing. Treating space as a sequence resolves numerous phenomena and suggests that the place field mapping methodology that interprets sequential neuronal responses in Euclidean terms might itself be a source of anomalies. So I think we talked about this a long time ago, where they have these different representations in the hippocampus, and it's not, some people have proposed it's Euclidean. Of course, I think we talked about Euclidean spaces, and machine learning, and how we kind of assume Euclidean spaces, and of course, the world isn't just Euclidean spaces; it's maybe more higher dimensional than that. And so, this is kind of where they're getting at. This whole idea of Euclidean spaces or Euclidean terms being a problem for understanding the actual representation. So, again, like in machine learning, Euclidean spaces are limiting, basically. Our model, clone structure causal graph, so this is CSCG, this is their model that they're introducing here, employs higher order graph scaffolding to learn latent representations by mapping aliased egocentric sensory inputs to unique contexts. So this is where we have this graph scaffolding, where things are built on a graph. You learn latent representations, you map. Egocentric means it's centered upon your body, and then you're looking outward. And so you're getting sensory inputs in an egocentric frame, and you're trying to apply those to unique contexts. And you have, you try to use this sort of information to learn these latent representations of the space. And then I guess you sample from those representations to guide you in your spatial learnings, I guess. So that's basically what they're doing. It's just, there's a lot of jargon here, but basically you're learning about the spatial world by, your sort of from your own point of view. And you have to encounter different landmarks. You have to build a representation of the space. And, it's all kind of this thing that you build from your experience, from your egocentric experience. Learning to compress sequential and episodic experiences. So remember I said episodic memory, or kind of different episodes in space. Like if you see a certain object from a certain point of view, that's an episodic experience. And the idea is you would put those together into a representation. You have different episodic experiences, and you build a model from that. You say, okay, this thing is here, this thing is here, this thing is here. And then you try to build a model around that, saying, okay, how do these things related in space? And then, of course, but you have to compress these because, of course, you learn many different spaces, and you only have so much hippocampus. Your hippocampus doesn't, okay, that's not true. Some people have, there's this classic study about London taxi drivers, and that, they're very expert at navigating their environment. And so the evidence for this London taxi driver study was to look at the morphometrics of the hippocampus and to show that it grows in taxi drivers, people who experience, episodic memories about the different streets in London all the time. They drive down them, they know the shortcuts, they figure out how it's represented, and so this is, actually leading to an enhanced sort of growth in the tissue of the hippocampus. But still, this isn't something that you can do sort of scale up based on the number of spaces that you have to learn. So you have to compress this at some level. So learning to compress sequential and episodic experiences using the clone-structured causal graph model yields allocentric cognitive maps. So allocentric cognitive maps are cognitive maps that exist out in the environment, not based on your own experience. And so that's the kind of map it's building, that are suitable for planning, introspection, consolidation, and abstraction. So all the things you would do with space, you would plan, how do I get from point A to point B? And it's not always the same way all the time. You have to think about, if there's traffic on this street or that street, or if there's like a detour that you have to make, or if that space is even, continuous or discontinuous. There's introspection. So when you go and visit these places, what do you learn about it? Do you learn that, the representation is incomplete, or what are you learning? Consolidation, of course, meaning that you consolidate all this information as you learn it, you consolidate it into a model. And then abstraction, so being able to take space and kind of, being able to think about it in different ways. So, we have spatial representations of, say, our friendship network, or spatial representations of, a place we've never visited before. There are a lot of ways we can abstract this out to other contexts. So while personal experience is important, we can abstract it to other contexts, and we can do this kind of generalization. By explicating the role of Euclidean place field mapping and demonstrating how latent sequential representations unify myriad observed phenomena, our work positions the hippocampus in a sequence-centric paradigm, challenging the prevailing space-centric view. So they're saying that, you have the space-centric view, which is where you have all these cells that code for different aspects of the space, and you build these representations of the space. What they're saying is that you can actually have a sequence-centric paradigm, where you take the sequence into account, the places you visit, and you sample from a latent space to build those sequences. So it's a different way of thinking about what the hippocampus does. And so, this is, of course, this may be, I don't know how controversial this is. It might be quite controversial, but basically, a lot of the previous studies on the hippocampus, we've sort of assigned a role in it for episodic memory. So, building these kind of series of episodes in a spatial representation, being able to do relational inference and fast learning. And so, this is important to understand. Our model suggests that place field maps need to be interpreted carefully because they overlay sequential responses onto Euclidean maps. So this is where, we have a lot of things going on here, I guess. And so, we have to kind of think about it a little, pretty carefully when we interpret the results of any one place field map. Directly characterizing the place field maps in terms of spatial and Euclidean concepts could be a source of anomalies, since the underlying phenomena are inherently sequential and dynamic. So again, we have this aspect, maybe a dynamical systems, where we're kind of building these spatial maps, but not thinking about how they evolve over time, or how information is acquired over time and integrated with them. In contrast, CSCG explicates how the learning of sequential contexts gives rise to spatial representations that an agent can use to drive behavior without explicitly representing location coordinates. So we don't want to have, there's this kind of assumption in a lot of spatial representations that exist that you have to build location coordinates for everything. Certainly when we do robotic planning or robotic navigation, you build these models that are sort of geometric and they have these coordinates, and you have to translate between the coordinates for different contexts. And, there's no real evidence that we're building coordinates in our brains. It's just that that's kind of the assumption, because we have this model, this Euclidean model, this model that we know from geometry that can represent the space of coordinates, but there's no evidence that we actually use coordinates. CSCG predicts the conditions under which place fields are expected to change in response to visible or invisible environmental changes when they do not, resolving a variety of phenomena with a simple principle. So this is an example here of a rat learning a cognitive map from sequential experience. So the rat is going through this maze, it's kind of exploring, it's trying to find its way back to the origin point. And so it's not a perfect thing, it's just kind of wandering around, finding its way out to the original spot, using these cues in the environment, these bricks, and kind of putting that together. And as it turns out, this is a sequence of egocentric observations. So as the rat looks out on the maze, it sees like this green block and this fuchsia block, and then it kind of moves, it turns and sees the green block and the orange block or the yellow block, and it moves up until it sees another set of blocks. So it's using these egocentric observations, and it's basically stringing those together to find this path. So this is where we have the sequence of egocentric observations, and at each point you can have a sequence of egocentric actions. So you see things in the environment. You see a series of things in the environment. Kind of when you give a set of directions to someone, that you often use this sort of approach of a sequence of instructions on how to get from point A to point B. So it might be like make a turn at this highway, or make a turn at the second mailbox. It doesn't really matter what the object is. It's just that you're observing the world in this sequence of sort of snapshots. And you can think of this as episodic memory, but then you can also think of this as a sequence of events that get strung together and coded into this allocentric map, which is a map of the environment. So you're taking these egocentric views of the environment, you're stitching them together, you're building this allocentric map, which is basically, if I look down at the space, what does it look like? I'm no longer in the space or like traveling in a straight line. I'm looking down at the entire space and I'm saying, what is my path? And I can recall that path. So, we can make an egocentric action at each point, but when we get to our destination, then we can encode this allocentric map. So that's basically what they're getting at with the sequential learning. You can have flexible splitting and merging of states. So this is where we have different sequential information that's put together in different ways. This is, of course, taking all the observed sequences, building a latent space, and then, putting them together in some order that makes sense. And so not every order makes sense. Some orders will make sense, some orders won't. And so being able to build a latent space and then sample from it allows us to build this first order Markov model, and then have this cloned model with appropriate merging and splitting of state D. So you can see that D is where you get A and B. They converge upon D. Actually, C goes to D prime, and then goes to G. D goes to E and F. And so that's basically what you're getting here. You're sampling in different ways. And so this is the CSCG graphical model. This is where we have an action state. We have the latent variables here. We have the discrete emissions of this latent variable. So this is sort of a Markov model of the whole process of learning spatial information. And then you have this observation at time step n. So you get this action, a latent variable, a discrete emission from that latent variable, and then an observation, the clones and emissions that are connected together in this way. So, this is where you're getting an emission from this latent space, and you're testing it out. And you're making observations after the fact. And you're putting all this together. Okay, so this is a 2D room with uniform interior. This is a set of mice that are kind of traveling around the space. You have the sequence of egocentric actions, these egocentric observations. So in spatial cognition, it's not only important just to have the sequence of egocentric observations, but it's important that you can rotate them in different ways so you can learn kind of, okay, if I'm here, then what is over there? And I can rotate my viewpoint. I can say, okay, if I'm headed in this direction, then I'm headed, this thing is over on the left. If I rotate around, this thing is on the right. So be able to do these kind of rotations as well is important. These are heading dependent observations. So your heading is, the direction which you're traveling. And so they do this. And they have this learn transition graph, which describes all of the different rotations, all of the different sequences of learning, and then mapping this to place fields, the firing rate in place fields. So they're able to take this representation here in 3 and map it to place fields. So this is where we have this learn transition graph among clones. Each clone's color represents the observation it's attached to. And then number 4 here is where clone activations as the agent navigates the room can be used to compute their place fields, which reveal the spatial locations they represent. So these networks then can be mapped to place fields, and you can understand kind of where these networks are in space or what they're representing in space. So yeah, they give a lot of methods here. This is, they did a number of experiments for this, where they kind of did a review of the list of experiments, their observed phenomena related publications. So basically this kind of goes over all of the, diversity of the field and how people are thinking about these sorts of components of spatial learning. So, you have this source O'Keefe and Burgess, where the experiment was changes in the geometry, and the phenomena observed was that place fields remap as determined by geometry. So that's one experiment. Then there was, of course, visual cue rotation by Muller and Kubi. This is where place fields rotate with cue card. Barrier addition, also Muller and Kubi. Place field disruption near barrier. So probing the place fields. Landmark vector cells. Place field remaps with respect to a landmark. This is Desmok and Kaniram. Linear track. This is where the place field remaps with respect to start and end of track. So you're taking the relative position of the start and the end of the track and seeing how the place field remaps with respect to that. And that was Sheehan et al. These numbers are in the reference list, so if you want to look it up later, you can. You have direction place fields. That was in O'Keefe and Burgess. Laps on a track. This is in Sun et al. 4 connected rooms. Duvel et al. 2 identical rooms. Fuchs et al. Hairpin maze. Derdikman et al. And room size expansion. Tanny et al. So these last 5 are, looking at how place fields are unaffected by certain features of the environment, but sometimes also affected. So, we have direction-specific repetition of place fields that expand or stretch based on the location with respect to boundaries, place fields being unaffected by closed doors, and event-specific rate remapping and lap cells. So there's a lot of stuff that they're trying to kind of integrate here. A lot of disparate findings, a lot of interesting things, but no theory and no, set of unified models. So this is actually an example of this CSCG learning in a simulated 3D environment. So here's an agent, and this is, of course, not with a rat. This is in in silico, where they're doing this. This is an isometric view of a 3D environment. So you have the agent here. You have the egocentric field of view. So the field of view moves as the agent moves. It takes in a part of this environment. Then you have this egocentric observation here. You have this discrete emission, which is the vector quantizer. You have another observation from the egocentric point of view, the vector quantizer. So there are these discrete emissions from this. From those, you build this learned transition graph, which is, of course, this, you move from state to state in this graph. So that's being able to learn in this 3D environment. There's latent transitive learning, which is where you have coverage of the 3D environment in an example episode. So your agent travels a certain amount of the environment, explores that certain amount of the environment, and then learns from that. So there's this coverage here. It's not the entire environment, but it's pretty good coverage, I guess. You end up with this learned transition graph that kind of maps out the space kind of crudely. So that's latent transitive learning. Then you have latent learning separate maps and disjoint environments. So you have the isometric view of individual 3D environments. So you have different environments. You can see that the agent is in here and they have a point of view, and as they move, it changes direction, changes its location. And being able to learn separate maps in different environments leads to these different learned transition graphs. So you have different graphs for different spaces. With shortcut planning, you have this space where you have some shortcuts in there. You have ramps. You have to go from like a source location to a goal location, and you have to find these shortcuts. So you have platforms and ramps that allow you to do these shortcuts. And the idea is the agent has to learn the ramp and the platform and their position between the source location and the goal location. So they do this. There's this initial walk that gets decoded, and then there's this execution of the inferred plan, where the agent takes the ramp to get on the platform to get to the destination. So it takes the shortcut successfully. Okay. And so, one of the things about their model is that it reproduces several place field remapping phenomena. So that table I showed you before gives you like an idea of what kinds of phenomena people are looking at. And this model, of course, allows us to reproduce these. And so these are the different studies that they have: O'Keefe and Burgess, Muller and Kuby, Sheehan et al., Sun et al., and Deshmukh and Kniram 2013. So all these are these different studies, and they've kind of replicated them using this model. So, we're able to do this with the model. And so they kind of finish up here by saying, making the statement that sequence representation can help explain puzzling phenomena that make spatial and temporal effects. So sequential contexts naturally explain the direction sensitivity of place field remapping reported in 27, which is this paper here. And so I think that is, when he was not important. So when the room is elongated, some place fields that were unimodal in the original room remap to produce two peaks, corresponding to two subcomponents in the elongated room. It was observed that the peaks were direction sensitive. The left subcomponent was active during the rightward travel, and vice versa. We tested CSCG for the same effects using the same settings as in figure 3a by plotting the fields conditioned on the direction of travel. In the horizontal rectangle room, rightward and leftward trajectories of the agent strongly activated the left and right peaks of the place field. So remember, these place fields are representing a certain place. There's a strength of activation depending on where the agent is. And so this is what they're kind of hitting. They're able to take these sequences, decode them, and then put them in these place fields. And the place field should be predictive of features in the environment. So this is exactly what they're finding here. And this is because only one of the sequential contexts that activate a clone occurs in a directional walk, which is a natural consequence of representing locations using sequential contexts. So this is a consequence here where we want to use locations and represent them using a sequential context. And so we have to kind of work around some of these issues with like directional walks and things like that. Okay, so this other thing here is CSCG can predict what kinds of environmental changes lead to remapping. So CSCG shows that environmental connectivity changes need not lead to place field remapping, even when the agent's behavior shows adaptation to the change. A phenomena that the researchers found puzzling. So in citation 31, rats are in a four-room maze where the doors connecting the rooms could be selectively locked to change the connectivity of the arena. The rat's behavior reflected that it recognized the connectivity changes of the environment, but the place fields did not remap in response to these connectivity changes. The authors found this lack of remapping puzzling and argued that place cells do not encode a topological map. So this is a kind of a controversial statement in that, some people believe that the place cells actually are involved in a topological map, and some people question that. So this is kind of an outstanding issue in the area of spatial cognition. However, CSCG showed that place cells can encode a global location in their activations, global topology in the cell-to-cell connectivity, and still not show remapping in response to the manipulations. So they did this. They demonstrated this by training one of their models using a random walk in an environment comprising 4 square rooms that are connected by two-way doors, similar to the experimental setting in this study 31. Each room had visual cues that distinguished it from the other rooms. The model learned the global topology of the arena and the transition matrix, and the activation of clones corresponded to locations, as in the previous experiments. And so then they tested this for 2 environmental modifications, where one door was locked both ways, effectively creating a blockade. And the second is all doors were locked in one way, allowing only an anti-clockwise direction of travel. So this is where you're kind of fixing in direction. So you're kind of either saying you're trapped in one room, trapped in one part of the space, or you're traveling in a certain direction and you can't go back. So this is so corresponding modifications were made in the CSCG transition matrix by modifying the connections appropriately, and planning routes in this modified CSCG corresponded to reported successful navigation. So the reason for the lack of remapping can be understood by realizing that the connectivity change blocked paths without any change in the visual cues. The blocked path affected only a few of the potential sequences that were responsible for that place field, a change that is too small to be reflected in the aggregate sequential responses. However, the connectivity change can lead to large changes in behavior, for example, in navigation between the 2 rooms. So this is where this is kind of this idea of it being emergent. So you have these, selected examples where they're blocked paths, and you have this connectivity change in the network, and it's not consistent with the visual cues. So but it only affects a few of the potential sequences, so you won't see this in the aggregated sequential responses. But you also get the potential for this sort of emergent behavior, where they can figure out a way to navigate between the 2 rooms that isn't necessarily obvious. So this, and then there's of course some testable predictions we can make with this model, and so they've kind of listed out every time you build a theory, it's important to have these testable predictions at the ready. So in this case, one prediction is that what controls how place fields change is not the rate of visual change, but the uniqueness of the visual context. So this is where, when you look at these place fields, how do they change? Is it the rate of visual change, or is it the uniqueness of the visual context? So they actually tested this, and they did this with an experiment with square rooms. They observed that the place fields in the checkerboard room were more expanded. This is because the same context repeats throughout the interior of the room, making it difficult for the learning to split into contexts for different clones. So this is a nice model. They can test different hypotheses. They can look at different aspects of spatial learning, which is a huge field that's kind of all over the place, and get some sort of predictive theory out of it. So there's a lot of stuff here. They mentioned Tolman-Eichenbaum machines, or TEMs, and that's in citation 51. This has many similarities to CSCG and inspiration. However, unlike our model here in the paper, TEMs do not learn latent graphs in alias settings like ours. So people have kind of hit on this idea before, but not entirely in a way that's effective. So I think I'm going to stop for now on that paper. Did you have any comments or questions?
No, I just, I find, I love this work in the way it makes neuroscience. It feels like it's revealing all the engineering behind neuroscience.
Yeah.
This is hard stuff.
Yeah.
And yet at the same time, it's explaining something that, to some degree, is seems so basic to us, or is something that a toddler is doing.
Yeah.
Right. Like it's anyway. Very, it's super super interesting, right?
Yeah.
You know, I didn't actually see how much is available to play with in terms of models that they have, or models that they make available.
Oh yeah.
But this absolutely, you know, what would of course be interesting is to see, you know, like this is very along the lines of the kind of stuff that like the neurorobotics platform from the old Human Brain Project was doing, with, you know, a kind of virtual rat, and then using spiking neural network model to do not necessarily some of their particular examples, but certainly just to do 2D maze or kind of route finding, you know, and like definitely simpler than these examples, but but again, something that was very much a combination of kind of like bottom up neural models that were helping explain a kind of high level behavioral task, which, you know, even though we don't think of that as like a super cognitive task, it's certainly something that, you know, all animals are engaging in, you know.
Yeah.
And not to distract from this paper, but there was a great what's the Build a Cell seminar series. Yeah. Like this week's Build a Cell was a really interesting Stanford, I don't know, like biophysicist. I mean, definitely started out as a physicist, and you know, like he shows a picture of him at a meeting with Feynman, like in like 1980 or something like that. But it gives a good example of that very simple single cell organism that can do, you know, all sorts of kind of real time behavioral action. And, you know, like we did with that, was it like Roman Brett, or maybe something we were reading around that time, where it was just like, you know, talking a little bit about, you know, that kind of organism, as well as just like ciliates in general. And, you know, like there's something cool about these kind of specific hippocampal models.
Yeah.
But there's also something really cool about like single cell organisms seem to be doing something similar.
Yeah.
You know, or is it? Well, anyway, it was a cool Build a Cell seminar this week.
Oh, okay.
Let me just say that I'll if I haven't, I think I shared it in Celsius. Because he also had a lot of microtubule dynamics with regard to, you know, that single cell animal going out and seeming to you know, feed, you know.
Yeah.
And like seem to know where food was, and or other animals were, and was grabbing them. Anyway, yeah, really interesting, really interesting paper.
Yeah. So you know, that's another point you bring up, is that like you can do spatial cognition. Maybe you need a hippocampus in mammals, but there are other models of spatial cognition. You have path integration in insects, and they don't have a hippocampus. Single cells can navigate, you know, to these taxis where they move towards a resource. They can navigate through their environment. Single cells within the human body can do that as well. based on just kind of coordinating different signals, chemical signals. So it's interesting how that, we need like a huge model of the hippocampus, and, we need all these different cells to navigate spaces, but other organisms do it with less, I guess. And, the question is why?
Yeah. Well, I mean, I think this is why, I love orthogonal, is that, that you're thinking about the full evolutionary range, right? And because trying to understand the hippocampus without understanding, its evolutionary context is probably, impossible, right? And there are so many. You mentioned insects. I think I mentioned Edwin Jammer, the Bristol prof, who had that paper on bird migration, and just had a simple theory about bird migration that had some cool computational stuff. Big question, but it does seem like there is all this complexity to the hippocampus, but then is that that complexity seem, potentially some people are saying like that complexity is then used for other cognitive tasks, right, outside of spatial, and like perhaps some of its complexity, or its something about its generalization as then allowed other parts of the brain to utilize it as well, right? I don't know, but and that we need to look back, so-called down the tree, even though maybe tree's not the best way to think about it.
Well, I mean, different taxa and how they're have a common ancestry, and then like how did they diverge? Like, we have, of course, fishes and birds and lizards, and then mammals and invertebrates. So how did they all do spatial cognition? It turns out they use kind of a semi-conserved mechanism. They have like the different lobes of the brain, and those have been refined, and they kind of use those. But then when you go over to insects, they use a totally different set of mechanisms. And in single cells, they don't appear to have like neurons. So and even neurons themselves don't have their own neurons. So
yeah.
Right. All right. So I guess that's good. So I guess to follow up on this, I know I kind of talked about the hippocampus. Maybe we're hippocampused out, but there's this other set of papers here. Reinforcement learning, never. Reinforcement learning development in the hippocampus. And these were two different papers. So this one is on sort of the development of time compressed sequences. We'll see what that's about. And then linear reinforcement learning and planning, grid fields and cognitive control. So it's this kind of reinforcement learning approach. So let's start with this one.
Do you mind, for my old eyes, do you mind blowing that up a bit?
Yeah.
Thank you.
There we go. Okay. So this is in, I guess, Science Advances again. No, it's actually in Science. So, okay. This is by Farooq and Dragoi. Emergence of preconfigured and plastic time compressed sequences in early postnatal development. So this is where we're looking at hippocampal neuronal ensembles and where they kind of first organize in development, and we want to know something about how this sort of emerges in development. So the abstract reads, when and how hippocampal neuronal ensembles first organize to support encoding and consolidation of memory episodes. So, we have these neuronal ensembles that sort of form in the hippocampus as networks, and we start to get this consolidation of memory episodes, and we don't know when that starts in development. We recorded electrophysiological activity from large ensembles of hippocampal neurons, starting on the first day after eye opening. So this is where we're in rats. We have this eye-opening event, and then they have to experience the world for the first time. So these are naive rats, and they have to navigate a linear environment, and they have to sleep to consolidate the memories. They let them navigate these environments, let them sleep, and then they wanted to see what kinds of things were learned from these ensembles that were existing at this point in development. So we found that a gradual age-dependent navigational experience and dependent assembly of preconfigured trajectory-like sequences from persistent location-depicting ensembles during postnatal week 3. So this is where we're interested in this early part of development, postnatal week 3. They found that there's this age-dependent experience, adult-like compressed binding of adjacent locations into trajectories during navigation, and their navigational experience dependent replay during sleep emerged in concert from spontaneous preconfigured sequences only during early postnatal week 4. So we're getting different things coming online as we're going from postnatal week 3 to postnatal week 4. And so we're actually getting adult-like spatial navigation by early postnatal week 4. So this is where it's kind of coming online, and we're looking at, as soon as they open their eyes, they get to experience the world, the visual world, and then, they have to kind of learn how to do this sort of like an adult. And so it doesn't take very long, apparently. Our findings reveal ethologically relevant distinct phases in the development of hippocampal preconfigured and experience-dependent sequential patterns thought to be important for episodic memory formation. So this is kind of goes over, so the hippocampus is crucial for relational binding of spatial locations and events into these spatial mental trajectories and memory episodes. So they're not talking about like the previous paper where they were laying out this theory. They're just talking about sort of the development of these abilities in ensembles that exist in the hippocampus. So we're not interested in theory. We're just kind of interested in what those networks are doing. So the rapid encoding and consolidation of sequential spatial experiences into memory episodes are achieved by representation of such trajectories within time-compressed hippocampal neuronal sequences during sleep or rest preceding and following novel experiences. So they're doing this test where they're looking at sleep, the effects of sleep on or rest before and after these novel experiences. So this is where, this is just sort of the way they've done the experimental technique. They just want to see what the effects of encoding are, or of, encoding a memory. The hippocampus undergoes a developmental critical period and functionally matures around postnatal day 24. So postnatal day 24 is kind of like between the 3rd and the 4th week. So this is where, again, they open their eyes at postnatal week 3, kind of at the beginning, which is about day 21, day 19, and then they experience this maturation of the network. But this is also a critical period. So, it has to be there. You have to have, if your eyes aren't open by then you can't acquire the information. So this is where we have this intersection of like these developmental events and then this critical period, and everything has to kind of be in place in order to get this result. But anyways, so there's this functional maturation around day 24. However, when and how the hippocampal neuronal ensemble patterns of activity across different brain states underlying the encoding consolidation of episodic like memory develop. So this is where, we're looking at these neuronal ensemble patterns across different brain states, and we're looking at the encoding and consolidation of episodic like memories. We're looking at the dependence on intrinsic developmental programs or prior structured experience. And this intersection of things, we don't really know how those all relate to one another. So this is in part due to the difficulty of simultaneously recording electrophysiological activity from sufficient numbers of neurons in freely behaving developing animals. So we need a technique that allows us to look at all these neurons and how they're doing things in a freely behaving animal, but also in this developmental period, this very specific period. We had a comment here. Okay, that's brain engineers, linked to brain engineers.
Sorry, I was just adding some links. I promised Trevor.
Okay, yeah,
I said I'd drop in before. Sorry.
That's fine. So previous studies of sensory motor brain areas have mapped how neuronal activity before and after experience relates to the developmental or synaptic connectivity and plasticity of that of the hippocampal network. So we're interested in sort of experience as sort of this threshold. Once you have experience, then there's this flurry of synaptic connectivity and plasticity, changes in this, and then we end up with our maturation. And so during this critical period, it's very important to have sort of be able to open your eyes and experience the world. And if you don't do that, then you don't acquire this sort of adult-like behavior. However, the hippocampus does not directly respond to external stimuli of a single sensory modality. Instead, it integrates multimodal sensory and locomotor information across different brain states during navigation, awake, rest, and sleep. So it's doing a lot of sensory integration as well. And that's something we didn't see necessarily in the last paper. We saw where they tested this on sequences of views, on egocentric views, and building an allocentric map. But another part of spatial cognition, which is important, is this aspect of footfall information. When you walk across a surface, you're moving your body, you're hitting the ground, you're counting your steps, and that's all part of the spatial information as well. You can also have auditory information that's important, sort of localizing sounds and things like that. So it isn't just about viewing scenes. There's a lot of information here that plays a role. And locomotor information especially, there's a lot of stuff going on when you're moving that play into this sort of memory, augment the memory and give it context. So this is where furthermore, episodic memory, thought to depend on the maturation of the hippocampal network, develops after sensory motor systems appear sufficiently operational. So your sensory motor system needs to be in place before you can experience spatial behavior or the hippocampal networks start to mature. Finally, structural maturation of the hippocampal formation is controlled by intrinsic signals from the stellate cells of the entorhinal cortex, and this is part of the hippocampus is part of the entorhinal cortex, and so this is there are all sorts of different things that communicate with the hippocampus proper in the entorhinal cortex. So this is where you have to have this bidirectional communication between the hippocampus and other centers in the brain, especially sensory motor systems. And we really need to understand the developmental stages of episodic memory formation within the hippocampus itself. So they recorded from the hippocampus from these different fields from 19 2 to 3 week old freely behaving rats during 30 minutes of their first ever exploration of a 1 meter linear track. So this is where they have this day one run. This is their first experience. Then they recorded preceding and following about 90 minute long sleep sessions. So you have the day one run, you record pre-run and you record post-run. So you get this sort of effect where you look at the network before experience, you give the rat experience, and then you measure post-run and see the difference. You also have these awake rest epochs that are, also measured. 14 animals re-explored the linear track on the same day. So this is day one, run two. So they do another run. On the following day, all animals re-explored the same linear track, or day two run. And this run session was flanked by sleep sessions. So again, we're looking at the first day, we're looking at the second day, we're looking at what happens before and after training. And then in some of the rats, they get a second bout in day one. So this is where they're kind of going and setting up this, almost like this, this type of data set where you have, almost like not quite a time series, but so they find out, they kind of get through some of the methods. This is the diagram here in A for the experimental design. These are the data here. They're recording from the neurons the first time run, and they're so for the first time run, they get this set of locations in meters, these representations that form. This is P15 through 16, P17 through 18, P19 through 20, P21 through 22, 23 for 24, and then finally the adult. So what you notice about these graphs is that the activity sort of becomes less. There's less variation across these different recordings. So you have this representation that moves from location 0 to location 1.5. In earlier days, or in the first days of training, or in the earlier parts of development, you get this sort of significant noise across the different neurons, and so it's hard for the mouse to find its way to a destination because it has all this noise in the network. And about day 19 or 20, the noise starts to lessen. This is probably due to some sort of synaptic pruning. And then, friendly for the adult, you get some noise, but largely this allows the rat to have a map of space, how to get from one location to another. There's this run and shuffle condition. So when they're shuffling, there's a lot more error in terms of centimeters per meter than in the running condition. So this just shows this over days of development for the adult. And you see that in development, you get this higher amount of median error for the shuffle than you see in the adult. But in the run condition, things converge down to the adult by day 23 to 24. So it's interesting. And then they look at theta sequences during the first time run, where they're looking at past, present, and future. And I'm not really sure what these graphs mean. This is just overdevelopment, comparing that with the adult. But I guess these theta sequences are more organized starting around day 23. And you see that that's very much consistent with the adult, whereas before it's very noisy, especially in day 16, day 18, day 19. And I assume that this is really kind of before any learning is acquired. So it's just kind of this line, and then you get this noise, and then you start to get this pattern, and then by day 23 it's refined enough to resemble the adult. So this is all kind of talking about this type of, these developmental aspects of learning. They propose a 3-stage developmental timeline for experience-dependent trajectory replay. You have stationary location depicting on days 15 and 16, partial trajectory depicting on days 17 and 18, and entire trajectory depicting on days 19 and 20. And so you have these different stages of replay during sleep in these different developmental periods, which are just 2-day periods. This is a graph of pre-run sleep versus post-run sleep. So you can see that you really have this transition at about day 23, and it resembles the adult. So this is, let's see. Here's some insight into what they found. So this suggests, and this result up here, this suggests that network preconfiguration does not emerge as a result of unaccounted sequential experience in the home cage before days 17 and 18, as an explicit sequential experience on a linear track did not result in a robust theta sequence and offline sequence plasticity until day 23 to 24. Previous day navigational experience did not accelerate the developmental emergence of age and recent experience-dependent plasticity experienced in day 21 and day 22 animals. Taken together, these results indicate that hippocampal ensembles exhibit reduced stimulus-related plasticity during the critical period. So this is where we have reduced stimulus-related plasticity during the critical period in hippocampal ensembles, including their experience in the home cage, and apparent contrast with heightened plasticity observed in the developing sensory cortex. So this was, I guess, a hypothesis that they had. Will we see changes in critical period changes in the hippocampus that we see in sensory cortex? And if you're familiar with sensory cortex, you know that sensory cortex really, refines itself over this critical period. And when you have like the sensory input, if you remove the sensory input during the critical period, you can really kind of mess up the animal for life, I guess is the way to put it. But in this case, we have this model of sort of critical period plasticity in sensory cortex, and we find that in hippocampal ensembles, we don't really have this. I mean, it's reduced compared to sensory cortex, but we do have some, critical period plasticity. So that's an interesting finding. I'm not really sure how to interpret it any more than that, but I guess, the idea is that there's maybe less of a dependence on this particular time period as opposed to sensory cortex. And so this suggests that the age-dependent emergence of time-compressed trajectory sequences depends on intrinsic instructive signals likely produced by the upstream developing stellate cell network of the entorhinal cortex. So this suggests that there's a sort of an innate aspect to training the hippocampal ensembles. You have this experience, but you also have this innate aspect where this other network is developing and it's kind of feeding information in. And so it's, maybe direct experience isn't always necessary to have, the development of spatial cognition, but I'd like to follow up more on that. It's kind of interesting. So they've identified distinct stages in the development of sequential patterns and neuronal ensembles in the rat, which we propose are important for the relational binding of events and episodic-like memory formation. In the first stage, the hippocampal network represents discrete locations explored in space, but lacks the ability to bind them into temporally compressed trajectories. So this occurs at 15 to 16, which is why we see this sort of immaturity. This is when the rats are confined to the nest, so we don't expect a lot of direct experience. The second stage is characterized by the emergence and gradual increase in the complexity of preconfigured sequential motifs, expressed during offline sleep or rest epochs, in an age-dependent navigational experience-independent manner. So this is where we have these, these sleep episodes where these things are being encoded as sequences, where the rat is kind of going over in their mind what they've learned that day and trying to put it together into a coherent set of sequences. And then this stage begins around 17 to 18 days and continues to 21 to 22 days when rats start exploring increasingly beyond their nest. So this is really dependent on when they leave their nest and get that experience, but this isn't as, I guess it isn't as sensory dependent or isn't as critical period dependent as sensory cortex. So this is kind of talking about a little bit more about plasticity. We propose that the coordinated development of time-compressed sequences during encoding and consolidation of sequential information by a stronger replay in sleep at days 23 and 24 in the rat represents a critical network mechanism that could enable the emergence of episodic-like memory function. So that's a good kind of summary of that. Quite a bit different from the first paper because we're talking about development. We're talking about kind of the sequence of things in development. We're kind of looking at the critical period versus maybe innate stuff that's going on. And I don't know if they've cracked the nut there. I don't know, but it's definitely all those things are coming into play.
No, just yeah, absolutely agreeing. Really interesting as a different take on the kind of developmental issues with the same region.
So the second paper then is this linear reinforcement learning, and this is Perret and Daw. So this is in Nature Communications, and so this is where we're interested in spatial cognition, cognitive control, but also this linear reinforcement learning aspect. So the abstract reads, it is thought that the brain's judicious reuse of previous computation underlies our ability to plan flexibly. But that also that inappropriate reuse gives rise to inflexibilities like habits and compulsion. So when they mean reuse of computation, they mean like, you're reusing the same mechanism over and over to find things. And I guess the idea is that you can benefit from that, but you can also, there are drawbacks to that as well. So there are a lot of habits and compulsions that are kind of fixed because the same computations are used over and over. And they're context independent, meaning that there isn't this, distinction between computations for like bad habits and computations for good habits. And this is just kind of a general thing. Yet we lack a complete realistic account of either. So what they're doing here is they're building on control engineering and introducing a model for decision making in the brain that reuses a temporally abstracted map of future events to enable biologically realistic, flexible choice at the expense of specific quantifiable biases. And so this replaces the classic nonlinear model-based optimization with a linear approximation that softly maximizes around a default policy. So in reinforcement learning, we have these policies that we try to use, and we try to maximize the behavior towards these policies, and the idea is that these policies match the kind of behavior we expect from the system. So this is of course, we have computational reinforcement learning, and then we have the kind of reinforcement learning we learn through experiments. And so the whole idea is that it's loosely based on what you would see in behavioral neuroscience, but it's supposed to give kind of a similar output. It's supposed to allow for learning and conditioning and things like that. So it replaces the class. Okay, we did that. The solution demonstrates connections between seemingly disparate phenomena across behavioral neuroscience, notably flexible replanning with biases and cognitive control. It also provides insight into how the brain can represent maps of long-distance contingencies stably and componentially, I guess, of having components, as in entorhinal response fields, and exploit them to guide choice, even under changing goals. So this is where we want to have these kind of, instead of having these like linear sequences, instead of having these other types of mechanisms and development, we're going to have these policies that kind of characterize learning and allow us to build these kind of flexibly changing systems where we can change the policy and the policy optimization. So that's what they're trying to do here. So they talk about the brain exhibiting a remarkable range of flexibility and inflexibility. A key insight from reinforcement learning models is that humans' ability to flexibly plan new actions and also our failure sometimes to do so in healthy habits and disorders of compulsion can be understood in terms of the brain's ability to reuse previous computations. And there are 5 references there. Exhaustive model-based computation of action values is time-consuming. Thus, it is deployed only selectively, such as early in learning a new task. And when possible, the brain instead bases choices on previously learned decision variables. This strategy saves computation, but gives rise to slips of action when cached values are out of date. So they give this strategy, and it's basically that you try to save time by deploying different computations selectively using this model-based approach. And then, you try to kind of use that for all different types of things. Now, it can save computation. It can save, the ability for you to do computation, so you might be able to do things faster, or you don't have to remember as much. But it also gives rise to different types of, a loss of different things like context or slips of action. So that's what they're getting at here. So this is the basic concept of adaptive recomputation. While it seems promising, this class of models, even augmented with refinements such as prioritized replay, partial evaluation, and the successive representation, which we haven't talked about, but I have a paper in one of the reading queues. There's a paper on successive representations that I'm not going to get into today, but is something, if you're interested, you should read more about. Has so far failed to fully account either for the brain's flexibility or its inflexibility. So, we have these models of computation, and the problem here is that, we can't say, like, if something is selectively flexible, we can't have a model of computation that is completely adaptive. You have flexibility that allows you to do a lot of things, but maybe comes at the cost of encoding certain information, or an inflexibility which comes from the need to save computation, but then doesn't allow you to explore new vistas. So that's the sort of trade-off here. And then, there are different types of mechanisms in these computational models that people have introduced to try to get this to work, but that's not, it hasn't, according to these authors, it hasn't been successful. For flexibility, we still lack a tractable and neurally plausible account of how the brain accomplishes the behaviors associated with model-based planning. So we're interested in model-based planning, and in the hippocampus, of course, we are using model-based planning to some extent. We have a model, we learn according to that model. And then, maybe if it's place cells, red cells, maybe it's sequential information, those models then will allow you to plan accordingly. So this is what they're getting at with model-based planning. Conversely, the reuse of completely formed action preferences can explain extreme examples of habits, such as a rat persistently working for food it does not want, but fails fully to explain how and when these tendencies can be overridden. And also many subtler, graded, or probabilistic response tendencies, such as Pavlovian biases or varying error rates in cognitive control tasks. So this is something for sort of computational psychiatry, and sort of thinking about it in that respect. How do you have these computations that overcome, say, bad tendencies or other types of errors in learning? It's interesting. I don't know too much about this field, so I'm not going to get into it too much. But they talk about this. In particular, we argue that the central computational challenge in sequential decision tasks is that the optimal decision at every time point depends on the optimal decision at the next time point, and so on. So if you were at a maze and you went from point to point in the maze, the value of going left or right now depends on, which turn you made at the subsequent junction, and similarly thereafter. So you're being locked into these possibilities with each move. And the idea is you want to be able to escape, say, the tendency of going down a blind alley every time you navigate the maze. It would be easy if you just had like a set of instructions and you just followed those instructions and hoped that you could make it through the maze. But of course, that's not the way mazes work. You have to discover, you have to explore as you go along. And then, of course, the idea is that you want to be able to make an optimal decision at each time point, each turn in the maze. And so that's what you're trying to enable with these models. The interdependence between actions is a consequence of the objective of maximizing cumulative expected reward in this setting, and is reflected in the Bellman equation for the optimal values. So they're using the Bellman equation to do that. However, it also greatly complicates planning, replanning, task transfer, and temporal extraction in both artificial intelligence and biological settings. So this is this sort of model where we want to be able to get rid of these biases in decision making. And we can use a lot of computation to do that, but it greatly complicates other aspects like replanning, planning, task transfer, and temporal extraction. So there are a lot of trade-offs here in some of these models. So how then can the brain produce flexible behavior? However, we lack a biologically realistic account of how this is implemented in the brain. Obviously, we're not implementing the Bellman equation in our brains. Maybe we do something that looks similar, but it's not biologically realistic. So the other aspect of this is because of the interdependence of optimal actions, exhaustive search seems infeasible for most real-world tasks due to the exponentially growing number of future actions that must be optimized for. So when we go through a maze, we're making decisions one at a time, and we're kind of optimizing for each step. But if you think about this in terms of all the options that we could make, we have to kind of make an optimal decision for a turn, and there may be 3 options for that turn. And then another turn that might be 4 options to consider. And you keep doing this over and over, and you can see how much computational power you need to kind of make that decision at every turn in the maze. One such proposal, which is perhaps the most promising step towards a neurally realistic planning algorithm, is the successor representation. And so this is in this paper, citation 16, which by leveraging cached expectations about which states will be visited in the future, we can efficiently solve a subset of tasks traditionally associated with model-based planning. So this is the successor representation, and like I said, I might do a paper on it next week, but we'll see. So they're doing this with Markov decision processes. So let's see what we have with the model here. So the model kind of goes through, so in Markov decision tasks like mazes or video games, the agent visits a series of states S, and at each they receive some reward or punishment R, and choose among a set of available actions A, which affects which state they visit next. So they have this Markov decision model, which they mention up here. They talk about that up here where I just highlighted. And then so, we have this optimal value V bar. Some state that is given the sum of future rewards, a series of nested expectations, or equivalently in the recursive form of the Bellman equation. Okay. Across all states, this results in a set of interdependent optimization problems, which can be solved, for instance, by iterative search through the tree of future states, computing the maximizing action at each step. However, in realistic tasks with large state spaces, this iterative nonlinear computation may be intractable. So, this is the kind of thing that they're doing here. They talk about model performance, replanning. Here's the linear reinforcement learning model in figure 1. We have this person stick figure. We have A and B. We have this coin here and this square, this red X. So for A and B, the model optimizes the decision policy by considering the reward and control cost, which is defined as the KL divergence between the decision policy and a default policy. So that's what we're doing here. We have a value versus probability of choosing A. We have the expected reward and the control cost. Then we have C and D, which is the model accurately approximates the optimal choice. We compared its performance on a 7-level decision tree task with random 1-step costs at each state to 6 pruned model-based RO algorithms, which evaluate the task to a certain depth of the tree. So that's here where we have this decision tree in C, and then this cost model in D, which we evaluate the cost of each model. And so that's what we have here. D1 is equivalent to the successive representation for the random walk policy, so that's D1 here, and uses average values as the weaves. Linear reinforcement learning achieved near optimal average costs. Y-axis is additionally cost relative to the optimum. Local costs of all states were randomly chosen in the range 0 through 10, and simulations were repeated 100 times. So I think I'm going to stop for now. I think we got the point of this paper. We're kind of walking through this decision-making model, and they're going to apply it to this this set of agents. So, do we have any questions or comments before we go?
So, it's a, I hesitate to say it, a simplification.
Yeah.
but it is the root of this paper a linearization and kind of a variational formulation of RL, without, again, without being, acknowledging that still leaves it very complicated.
Yeah. I think you're. Yeah, it's sort of a variational approach to reinforcement learning.
The use of the KL between the model, this is figure 1A, B blurb, the text, model optimizes decision policy by considering the reward and control cost, which is defined as the KL divergence between the decision policy and the default policy.
Yeah.
And like before that, there was some talk of we're linearizing this too, these typically when I've seen these variational reformulations of problems, like the linearization is also an important part of I'm making what was previously an intractable problem tractable.
Yeah, basically.
And again, I still think that leaves it really complicated.
Yeah.
but that seemed to be their goal. Yeah.
Yeah, I think so.
And for sure, like you said at the beginning, there's a lot of the usual suspects in computational psychiatry and the references from 1 through 5. Right?
Yeah.
Yalniv being one of those in the reference 1.
Yeah.
So, and so I don't think Dayan is that. Do you say Dayan?
I'm not sure.
Dayan. How do you pronounce
that? Dayan.
Anyway,
Yeah.
Dayan.
Yeah.
Anyway, Dayan's in there, and Botvinnik, of course. And Ida Momenijad.
Oh, right, yeah.
Yeah. Being, all. I don't think they call themselves computational psychiatry people. Y'all really is, but calls herself that. But they the methods are all completely, completely overlapping in terms of what they do.
Yeah.
So it's super interesting. It's this kind of thing, and again, it was the presentations that I found to be the most, edifying. Is maybe Ryan Smith. So definitely a computational psychiatry person. In that he's in psychiatry, like he's in the psychiatry department, I think, but he's a big active inference, he's the one who helps in some of his tutorials and some of his very explaining the differences between a whole number of computational models, all in an RL context, but then all as trade-offs between, like you think, exploration and well, I usually see it as exploration and exploitation, but I'm not sure what the exploitation is in this context. But,
well, I think it would
you
be,
know what I mean?
Exploration and fixed behavior, or
right, and it was nice to see where active inference it was much clearer with his I think active inference talk most recently, or active inference institute talk most recently, that it was the best explanation I'd seen in terms of what active inference is as a trade-off in this kind of RL context.
Yeah.
And
whose talk was that again?
Ryan Smith.
Okay.
So he's like he was definitely, I want to say, a postdoc of Friston's, postdoc or visiting faculty, something like that. Excuse me. And like he did a tutorial like a couple years ago. Like I think that the paper is called like a tutorial on blah 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 blah. I've like I shouldn't say blah blah blah. Was it dynamic causal modeling? I think it was. He did a tutorial on dynamic causal modeling, which is potentially more brain imaging work, but the really interesting thing about dynamic causal models, as it does relate to brain imaging, is how you can bring the experiment and particular brain network in this very clear model-based way into analysis. But his most recent talk, it's Brian Smith. He's at the Laurent Institute of Brain Sciences in Tulsa.
Okay.
Because they obviously got a lot of money recently, or like somewhat recently, and they hired a whole bunch of great people. It's good to sit on all that oil money.
Yeah.
But yeah, yeah, I'll have a look for that talk, because it's like we there was a guy at Stanford. There's an RL group at Stanford that ostensibly is working. They're calling them world models.
Yeah.
But it seems like the secret sauce is actually a lot of the same discussions that Ryan Smith's talking about in terms of trade-offs in the RL machinery, right? Because certainly some of the early parts of this paper related to the just basically what Sutton what was Sutton trying to accommodate with temporal difference RL. Sorry, isn't that what he called it?
Yeah.
What do they mean when they say TDRL?
Yeah. Temporal differencing.
Yeah, and it's always, I mean, related to this in general, and of course they mentioned Sutton, of course, in the references, but what's it called? Amy, I think, is the Alberta, or sorry, Emil. I'm trying to work out the acronym, but the Alberta plan.
Right.
So Sutton's group, the now, I don't want to say disbanded, but now defunct DeepMind Alberta, which is, they've been released and set free.
Yeah.
They're at that Alberta plan has a whole bunch of like, we're interested in these things in kind of RL, and it's a lot of these same kind of, sometimes it looks like it's fine tuning, but like I think if I understood all the issues better, I would see for its its true depth.
Yeah.
But super, it seems really related to this work in the sense that like it's related to it's almost like a kind of reformulation of the problem. But again, it's like it's solving issues that I just don't understand deeply enough to see it's, you know, like why it's so important, or why it's more than just computational tractability, I think is what I'm trying to say.
Yeah, I think that's true. I'd have to go back. You know, we should probably go back to Sutton's work on this, because
you know, I mean, yeah, like the reason I always for that same problem, right? It's just like if I really understood it as deeply as he does, then I'm sure I would get why all those points are important.
Yeah.
But until then, I'm just going to keep reading that list and trying to make a little bit of progress. But it would be good. I mean, again, it would be good to bring these, you know, because it's like, well, you know how groups are. They get a little in a rut with their own vocabulary and things like that.
Right.
But they're actually talking about the same things. You know.
Yeah.
And so this is a nice, or at least, you know, sort of while you were talking, I was checking the references as they were kind of going past in the text you were reading.
Yeah.
And yeah, like this is the, you know, the confluence of multiple research groups, or, you know, like you're seeing multiple research group work kind of come together and be well, more than merged, but you know, built upon.
Right.
And it would be great to see where, again, like world models and perhaps even every good regulator, could plays into this, you know. Yeah.
All right. Well, that's great. Yeah, we should follow up on this more. But yeah, this is great. Been a great discussion. Trevor, did you have anything to say before we go?
You know, you mentioned that you don't have too much, I guess, exposure to these ideas of model learning and reinforcement learning and whatnot. The only reason I unintentionally have stumbled into this area in the past 2 days.
Oh, okay.
So there's a paper you might find interesting. It's really more of an attack paper, but it was just a lot of the history of some of, you know, there's a really deep focus on dopamine. Oftentimes, it wasn't emphasized too much here, but often in a lot of these reinforcement learning paradigms, dopamine's like king. And anyways, so there was just an interesting paper I came across where they were challenging that, and like in a lot of these paradigms where like intracranial stimulation is used, it turns out that when you're trying to use that in the context of learning or to incentivize behavior, you're not actually replicating what the brain is intrinsically doing. It's you are creating a supraphysiological stimulus that in of itself is what is guiding behavior. And anyway, so this is, you know, the overlap is minimal, but if you have any interest, I'd be happy to drop a link. But sorry, I know it's not terrible.
Definitely drop a link. I think I know the paper you're talking about, or certainly one that sounds like that. Because I think certainly the original paper that we might be all thinking about is like the like 1997 Reed Montague, and I'm trying to think of, was it
hyperscanning, or was it different?
Anyway, he was definitely trying to relate, you know, a neural model to reinforcement learning.
Oh, okay.
and you know, and I say like, and I kind of follow Reed in particular because he's the head of brain imaging and yeah, I think like human neuroscience lab at Virginia Tech, my hometown.
Okay.
But what he's doing there, so he's one of the only groups that collaborates with neurosurgery to do neurotransmitter level detection in awake humans.
Okay.
So and so they've made specific devices to track neurotransmitters, potentially with a task, you know,
yeah.
while I have the skull open. Anyway, but I do I'll check the paper. Super interesting, Trevor.
Yeah.
Because I'll check the paper in terms of like I seem to remember somebody recently having a paper on like, no, it's not like the Montague. I can't remember who. It was somebody big in machine learning that was the senior author on that 97 Montague paper. But
okay.
Anyway.
Yeah,
Cool
I'd
stuff.
love to see that paper. It sounds interesting.
Yeah. Okay. So let's drop links. I mean, I put links in the Jitsi chat, but let's drop links in Slack so that they don't go away when we close this. And yeah, we'll follow up on the put them in computational psychiatry. Yeah. And I think that'd be really interesting because you know, again, like it is really hard to say what's going on in the brain,
right.
and you know, Reed, it's cool that he does. He supports kind of, you know, machine learning computational psychiatry as well as does neurosurgery and builds equipment to actually do the measurements in humans in a way that is the most comparable with works that's done in non-human primates.
Yeah. All right. Well, yeah, thanks for all that. Yeah, well, we can continue the discussion in computational psychiatry. I look forward to seeing some of these links.
Cool.
All right.
Yeah, thanks so much.
Yeah.
Talk to you soon.
Talk to you
Take
soon.
care, Joe.
Take care.
Bye.
You too.
Bye.
Bye.